It is my incredible pleasure to welcome Dr. Sarah Pressman. She's a professor of psychological sciences at the University of California at Irvine. I have known Sarah for many, many years, and so it is, really great to have her. She's also the associate dean in the Division of Undergraduate Education, and her research focuses on the interplay between stress, psychological well-being, which is why she's perfect for this, and health, with a focus on physiological processes and behaviors that underlie these associations. Dr. Pressman received her PhD from Carnegie Mellon University, where she published a seminal review and meta-analysis in Psychological Bulletin on positive affect and health, and really, spearheaded and kind of, was a pioneer in this area around positive emotions and physical health outcomes. She later did her postdoctoral fellowship at Pitt in the cardiovascular behavioral medicine training program with Karen Matthews, and then was an assistant professor at the University of Kansas before staying at UC Irvine. She is, a great researcher, great speaker, and been a great collaborator over these years, and for me, a great friend. So I'm really excited that she's here. She'll be giving a talk called Improving and Expanding Wellbeing Measurement to Further Our Understanding of Biological Impacts. Sarah, I guess you can share your screen.
Yeah, can you drop yours so I can share mine? Guess I need to press screen share first. That would help. All right, can you guys see that okay? Thumbs up. All right.
Oh, yes, but Sarah, we're looking at your comments too.
All right, so how do I turn that off? I tested this too ahead of time.
So I think you can share it and then hit the thing that says switch. There's like a switch button once it's shared.
Oh. All right, let me try that. So do I screen share first?
Yeah. Start the presentation.
Okay.
And then see how it says swap displays up in the left corner?
You're amazing. Thank you. I knew there was a reason.
That looks perfect.
Thank you, Eric.
You're welcome, pandemic.
All right, so thank you all so much for being here to listen to all of the panelists speak today. I'm so honored to be part of this panel of people who I completely admire. Some of my favorite people, some of my favorite scientists put this together and our speakers today. So it's just really exciting to be here. I'm going to invite you all to I think I'm like the odd one out of these last of the four speakers, in that they've been talking in such a complex level about biological measurement and really trying to understand these mechanisms that help us, get well-being under the skin and explain health. And I'm going to invite you to kind of flip that. And I'm going to talk to you about how we should also be thinking at that level of complexity with our psychological measurements. And so, like Eric mentioned, I've been doing this work for almost 20 years. This was the mechanism slides from way back when in 2005 on how we imagined positive affect could, get under the skin. Obviously, this was before mitochondria were in the conversation. This is before genomics were in the conversation. But we had these ideas that positive affect, either via main effect processes, were altering our health practices, which could lead to immune behaviors. So that fits into all of Ted's discussion on restoration, all the stuff on allostasis we've been talking about fits into the discussion of stress buffering, which I'll get to in one sec. But, autonomic function, such as parasympathetic and sympathetic activity, hypothalamic pituitary activity, and that these things are going to get under our skin to impact immune and cardiovascular health. And of course, relevant to Steve's focus on relationships and Ted's discussion of that as well, we always thought social ties would play a big role, especially given what we know about the importance of social well-being for psychological well-being. These things are extremely closely intertwined. And also, when we think about allostasis and the response to stress, we know relationships and social resources play a big role there. And so our stress buffering model, which has a lot in common with, for those of you familiar with Bart Fredrickson's undoing model, just this idea that positive affect can undo stress responses, leading to this restoration, just like Ted was talking about, but also that it may play a role in different places, for example, changing how we perceive stress, changing our reactivity to it, and leading to obviously a host of different psychological and physiological changes as a result. And so, we clearly know a lot about this, and it's actually really exciting to be here today and, hear all these innovations from the physiological side. But the thing that's really confounded me is, given how much we know now, and given how much we see that, these things affect physiology and stress responses, we should be seeing health benefits, right? We should be seeing that if we try to make people have more happiness, Steve mentioned eudaimonia, if we try to bring life meaning, these things should lead to better disease outcomes. They should lead to us living longer, surviving, chronic illness longer. And unfortunately, this doesn't seem to be the case at this point. And so this is a review that I did with the fabulous Judy Moskowitz, who spends her life trying to create amazing positive affect and well-being interventions to improve health. And we're just not seeing benefits. And also this was done with my wonderful former grad student, Brooke, who's now faculty at Chapman. And so, we're seeing some benefits to psychological well-being when we do these interventions. We're certainly seeing some behavioral changes. People are exercising more, behaving better, but we're not seeing the health questions yet. And one of the things that this really led my lab to ponder is, what are we missing, right? What's going on? What are we not understanding that's making this not work in interventions? And what this really led us to think about is the affect part of it. So we've heard a lot about different well-being constructs today. I'm going to focus today on positive affect just because that's what my lab mostly focuses on, which is essentially, positive emotions like happiness, excitement, feeling calm. They can be measured at a short-term level. They can be measured as a trait or a disposition. And, maybe what we really need to be doing is thinking more about certain nuances of positive affect or other well-being constructs. Steve mentioned the hedonia versus eudaimonia difference. And maybe if we do that, we'll be able to understand this better and perhaps develop down the line, not just a better understanding of how this is all working, but maybe better interventions as well. And so this is discussed extensively in a brief sort of theoretical review in Current Directions that my former grad student, Marie Cross, who's a faculty member now at Penn State, wrote just a couple of years ago, where we really talked about not every possible nuance, but a number of them that we thought can make a big difference relating to immune function like we're talking about today, but also potentially other health relevant outcomes. So more than just immune function. And so again, this is not inclusive, it's not a complete list, but it's probably what I have time for today, I hope. Which is that we really started thinking about a number of moderators, mechanisms, and measurement issues that may make a difference in why sometimes these things work and sometimes they don't, in terms of a well-being association between psychological well-being and physical well-being. And so the different things we talked about a lot in this review were issues of things like scale type and the kinds of assessments within that scale. What kind of questions are we asking? Issues of how well-being is measured, whether or not it's measured the typical way, which is just asking people on a piece of paper on a computer via self-report about how they're feeling, versus other alternative methods like looking at the language that they use and coding those words, asking informants about how they're feeling. And what I'll be talking about today a little bit is facial expression and whether or not that's a potential source of well-being information. I'm going to discuss, because we focused on this, the importance of moderators like culture and how that might make a difference in how these, psych and health variables interact. We'll talk just very, very briefly about some, nitpicky methodological issues like affect variability and timing. And hopefully by showing you just some brief examples of each of these, you'll get a sense of how, I know this is a lot, it's probably just as complex as some of the things we heard with how mitochondria work and how these genomic factors are working, but I think it's valuable. And so I'm going to just try to, my goal today is just to show you a quick example of each of these and to invite you to consider how this might better help you understand well-being in biology and maybe shift how you analyze your data or design your studies in the future. Okay, so I'm going to start off with culture. and the very famous Pittsburgh Common Cold projects. If you're not familiar with this seminal work by Sheldon Cohen, who was my graduate mentor, this is one of the few immune outcomes where we can actually give someone a disease. There's not a lot in health psychology and medicine where we can infect someone an illness, but the common cold is one of those things. And so in these studies, what Sheldon has done over and over again, there's about 5 of these now aggregated into a common data set, is he selects a healthy group of individuals who have never been exposed to a virus before. It might be a cold virus. He's actually looked at coronaviruses, coincidentally, before obviously this coronavirus, flu viruses. So everyone is virus naive. He isolates them in a hotel, but before that finds out about their stress, their well-being, their social relationships, puts droplets of the virus that's of interest into their nose, and then objectively examines how sick they are by looking at things like how congested they are with something called mucociliary clearance time. So how long does it take a drop of dye to make it from your nose to the back of your throat? Or even more fun, collecting all the Kleenexes that people use and measuring the mucus in them so that we can objectively see how sick people are and if they're objectively showing cold virus symptoms. They're not just saying that they're sick. We know that they're expressing that virus and showing symptoms. And so in Sheldon's work, he's fortunately examined the question that we're all interested in today, which is this question of positive emotional style, which is just in these studies, an average of the experience of positive emotion over a few weeks. And he's shown repeatedly in more than one study that your probability of developing an objective cold when you're exposed to it increases the less happy you are. And so what you're seeing here is that people who are less positive coming in, that's the blue group, had about a 34% probability of catching an objective cold and expressing it, versus people who are coming in with the highest level of positive emotion. And you see that with them, only about 19% of them were getting sick. And so this is amazing. This is an important finding in our understanding of emotional well-being and physical well-being, but we need to think about nuances, right? And so for me, one thing that jumped to mind is this issue of culture, because as an affective scientist, we know that there are lots of differences in how people perceive emotion and experience emotion. And so just as a few examples, people idealize affect differently. They want to feel different things based on their culture and the country that they live in. There are cultures that are fearful of happiness and are very high in something called the fear of happiness scale. And certainly in this country, there are norms, right? There are norms that encourage us to report or display positive emotion, to express our happiness, even when we're not necessarily feeling it. And so we looked at this recently using an aggregate of the 5 Pittsburgh cold studies to look at whether or not positive psychological factors equally predicted this protection for both African Americans and European Americans in this study. And this was work done with my 2 grad students, Cameron Wiley and Kennedy Blevins, as well as Sheldon. Okay, and here's what we found. So really, really interestingly, and I think we were dumbfounded to find such a robust effect. What you're seeing here is that our African American sample, which is about almost 300 people, about 250 people out of the study, that there was essentially no benefit of being high in positive emotion. And so whether or not they were high or low, it really didn't make a difference in terms of whether or not they got sick. But for European Americans, this is the effect that Sheldon's published on multiple times, right? People who are higher in positive emotion are less likely to get sick versus the people who are lower in positive emotion who are more likely to get sick. Now, positive emotion in this case wasn't necessarily harmful, but when we looked at the same analyses with measures of a different well-being outcome, specifically we looked at measures of self-esteem and self-assurance, we saw a harm effect. And so in this case, what we're seeing here is that African Americans, as they report more self-esteem, they're actually more likely to get sick. And then the opposite is true, which is what we would expect from the broader literature for European Americans. So they were actually protected by self-esteem or high self-assurance, which showed the exact same pattern. And, we find this really interesting, as far as mechanisms, we didn't find any obvious mechanisms explaining this really interesting effect. I'm happy to talk about that during the question and answer period of what we looked at. But my student Cameron Wiley is actually being funded right now by this very well-being network to look at this further in MIDAS and really try to understand it. both in terms of mechanisms as well as whether or not it replicates in other health outcomes alongside my colleague Dwayne Williams, who's an assistant professor at UCI. And you can see that we all belong together because we are all crossing our arms in this way that apparently at UCI you have to do in a photo. Okay, so let's move past culture, and we will look into this issue of moving past self-report. And I'm not dinging self-report. I use self-report all the time. It's a great tool. But for a long time, not just me, but many people have argued that this is something we have to be really careful about when we're studying well-being because of this exact social desirability thing that I was talking about with the cultural findings, which is in certain countries, we're very pressured to say that we're happy or to not say that we're depressed. And of course, there's all sorts of other problems with self-report, like response styles and rating scale issues, et cetera, et cetera. And I'm not the first person to talk about this, but we talked about this in a recent, not a recent review paper from about 6 years ago with the late amazing Dr. Ed Diener, my grad student Desi Delgadillo, and my former grad student Johnny Hunter, who's at Chapman. We talked about this, and essentially our conclusion was like, it's not that we should stop using self-report, it's that we need to take a multimodal approach because we need to know that these findings aren't just happening because of the common mode of self-report, and that, can we see some generalizability if we look across methods? Are we seeing the same picture if we look at non-self-report? And it'll give us some confidence that, these positive emotion health effects are real, that, it's really affecting physiology in the way that we think it is. And so this is one of the paper that was tacked onto the background readings, but my former grad students, Marie and Amanda, and also former UCI grad student Kate Leisure, and I wrote a conceptual review on this, really thinking about why smiling might be related to health. And, I think the easiest thing to think about is just that smiling can represent positive emotion, right? And so all the reasons that we showed in the first slide on why positive emotion can lead to better health are why we're seeing these benefits. And indeed, we see this in a lot of different studies. If we compare people who are sincere smiling to people who are fake smiling, and so sincere smiling is basically easy to identify. Sincere smiles have this orbicularis oculi activity, which is just wrinkles around your eyes. Fake smiles or social smiles, those don't. And so what we see is if we code for that in photos, looking for these markers of sincerity, that these individuals who are smiling more sincerely, which should reflect positive mood, and Ken Sheldon has a great paper on how Duchenne smiles are this honest signal of people being more dispositionally positive. And papers have shown that, people who do these kinds of smiles, they live longer, they survive illness for longer periods, things like renal disease and heart disease. And in a study that we did at UCI, we showed that people who are smiling more in their student ID cards are more likely to take care of themselves. They go to the health center for preventative reasons. And so this is great. This is convergent validity that says, okay, even when we're looking at non-self-report, we're still seeing the same kinds of patterns of health benefits that we would see with self-reported positive emotion. So how this is happening, it's probably not that complicated in these kinds of photo studies where we're documenting just how happy you are in a photo. It's probably something along the lines of trait positive affect, predicting smiles. This is leading to maybe better social relationships. People who smile more tend to have better social relationships, and maybe that's leading to greater health. Maybe it's just, this is going both ways, and it's just trait positive affect predicting health, like we've been talking about. There's a whole other thing on social desirability that I won't talk about, but I'm happy to take questions on how social desirability fits into this. Now, that being said, I entered into a new realm of studying smiling, and this is, instead of looking at smiling as an indicator of positive affect, we can actually use it as a predictor of positive affect. And this is by relying on something called the facial feedback hypothesis, which is the idea that if I make you mimic a facial expression, such as smiling, that will elicit that emotion in, cognitively. You'll experience that emotion when you're faking an expression. And the classic work does this without people even knowing that they're making this expression. There's great cover stories. So in this paper with my former grad student, Tara Kraft, we told people it was a multitasking study, and they had to hold these chopsticks in weird ways that simulated either, they didn't know this at the time, but simulated either a fake smile or a Duchenne smile while they were going through horrible stressors, like something called the mirror task, where you have to do a difficult time dexterity task, or everybody's favorite, the cold presser, where you have to submerge your hand in near freezing water. That's obviously very painful and it elicits a stress response. And what we see is, like we would expect with positive emotion, individuals who were in the Duchenne smiling group had the lowest stress reactivity as compared to the other groups, and also recovered faster than individuals who were fake smiling. That's the sort of lighter green color, turquoisey color. And even more so, obviously, than the people who are not smiling at all, which is the neutral group. And so we're seeing a nice reduction in reactivity and improvement in recovery just from smiling. And very cool, we were able to conceptually replicate this more recently by having people do these same kinds of smiling manipulations while they were getting a shot. And so this is just one of my students. This is from when I was at Kansas, putting chopsticks in his mouth as the nurse prepared him for a shot. This is just a shot of saline, but it's the same gauge of needle as a flu shot. So essentially it's a fake flu shot. And what we see is that people who are smiling had really excitingly a 40% reduction in how much the needle hurt, which is a really incredible effect size. So we were kind of amazed by how powerful this was. But more from the perspective of the talks today on physiology, this translated into lower stress reactivity and improved stress recovery in the people who were smiling. This is the Duchenne group versus the non-Duchenne group here, who both were better than people who were not smiling. You can't even see that line because it overlaps so much with people who are making a grimace. Grimace is an interesting contrast because grimacing does activate orbicularis, but it also activates muscles associated with negative emotions. And so we think that basically canceled out any of the physiological benefits there. Okay. So just returning to this facial feedback hypothesis, because I think it's kind of a fun idea. You know, we're moving smiling away from being just this indicator of positive affect to smiling as a predictor of positive affect. And this is controversial. There have been some failed replications. I'm happy to talk about the status of facial feedback and you know, some work that I've done on this as part of like a multi-site replication. But you know, what we're saying here is that smiling is triggering positive affect, and that is potentially directly leading to these physiological benefits, as opposed to necessarily even involving positive affect. And that's actually especially true in our studies where positive affect wasn't a mediator. And so this really led us, and this will be where I go deep into the biology side, really thinking about some new biological mechanisms. It led us to really thinking about, are there some, you know, direct connections between the facial activity in our faces and certain physiological processes that could explain these kinds of stress benefits. And I believe Steve hinted at this. I think it was Steve who talked about this in his talk. But this idea that the vagus nerve is connected with a lot of facial muscular activity. And there is, in fact, something called the oculocardiac reflex that you may not have heard of before, but maybe you've seen this in like a movie or a TV show about a medical emergency room. And this is just a reflex where if you push on your eyes, it actually results in an increase in vagal activity and a drop in heart rate. And so sometimes in emergency rooms, they'll kind of push on your eyes to try to calm your heart down. And so what this says for us as a possible mechanism is that activating these afferent trigeminal nerves could have direct autonomic benefits via activating parasympathetic function. And it really gives us this cool new physiological pathway for how well-being can be having those recovery effects, because it might not even involve emotion. This could be just direct physiological pathways between a positive emotion behavior and this physiological benefit. Okay, so cool new sexy biological pathway to consider. And again, we wouldn't have discovered this if we hadn't really started thinking about smiling as a way to test positive emotion. And it led to a whole new pathway of positive emotion expression, smiling, actually predicting physiological change, which is sort of an exciting new direction for our lab. And I will say in Marie's dissertation, she did find some evidence for this oculocardiac reflex, and she showed in her studies of pain that orbicularis oculi activation, again, those muscles around the eyes, did fully mediate the connection between smiling and RSA recovery. So hopefully there'll be some new studies that look at this, but it does look like a promising way for us to understand how positive motion is leading to that vagal benefit or vagal tone increase and stress benefits. Okay, so I mean, I think this is cool just from the perspective of, you know, what's the function of smiling and what's the function of emotion. This is a great paper by Azim Sharif and Jess Tracy from a few years ago, where they were trying to understand the hypothesized physiological function of all sorts of different emotions. And what you can see is under happiness, they just wrote research needed. We didn't really know. We knew that from a communication perspective, it can help with social relationships. It can help us, express this lack of threat. But I think what we've pointed out is this potential physiological benefit, which is, if we want to use smiling as a way to communicate a lack of threat, maybe we also need a way to calm ourselves down quickly. And this is giving us a pathway for how to do that, which is kind of exciting. And I will just say it also helps us understand a lot of old work in the laboratory where we see that people smile a lot when they're stressed, when they're embarrassed, when they're uncomfortable. Even grimaces look a little bit like smiling. And so I think this kind of helps us explain this natural human behavior in a cool way. Okay. So now I'm going to turn to something totally different, but this has been an obsession for me for many, many years, which is the issue of thinking about how we measure positive emotion and how arousal may be a really, really key piece of the pie puzzle in terms of how we understand why it's affecting physiology and well-being. Okay, so, one thing that's true about a lot of emotional well-being research is that unlike negative affect research, which is highly differentiated, people really separately study anxiety, they separately study, depression. They very rarely just study negative affect as a whole, but in positive affect, this is really what we tend to do. And worse, we tend to do it with these very highly aroused measurements like the PANAS and measures of vigor in the palms. And the problem with this is that there are different physiological correlates for high arousal versus low arousal measures of positive emotion, right? When we feel excited, this is an arousing emotion. When we feel calm, it is not. And so not only is that an issue, but it creates all sorts of problems, right? Because if we're averaging across all of these, it means that we could be missing some interesting nuances when excitement maybe predicts differently than calm. If these are working in opposite directions for whatever biomarker you're interested in, then you're going to potentially obscure that effect. And of course, you might not be measuring the kind of affect that matters if you're focused on this average, because maybe whatever you're trying to manipulate or whatever you're looking at as a correlate isn't associated with the biomarker that you're interested in. And so I've really been diving into, let's look at this more closely, especially because of the popularity of the measure called the PANAS, which heavily relies on words like active and strong and is entirely activated. So all of the emotions that are tapped in the PANAS are high arousal ones. And this is the most popular measure of positive affect used in health research by far. So what do we see if we do try to separate these out? Is it useful? Well, across numerous studies, we see that high versus low arousal, it does make a difference. So high arousal positive affect, feeling excited and vigorous, predicted longevity. Feeling high arousal but not low arousal predicts the common cold in Sheldon studies. Surprisingly, in some work on sleep and stressed out undergraduates, we saw that high arousal positive affect was associated with sleeping better during high stress periods, but not low stress periods. And as we heard from Ted, we see that high arousal, and he didn't present this, but he did look at in the PANAS, it was at the highest arousal stuff that predicted, and it was that predicting the wound healing. And I saw someone ask for the reference for this, but we're all talking about wound healing, so I'll put it into. This is just that finding that people who are low in positivity are healing at about a 20% lower rate than people who are higher in positivity, who are about 50% healed by 2 hours after being wounded through the tape stripping procedure that Ted talked about. And so again, this is all high arousal. So it's really pointing at this high arousal piece. And this has led my lab to really dig into what is high arousal and asking the question of whether or not past findings connecting high arousal like vigor and the PANAS are really due to more emotional items versus these energy and health-oriented items like what I mentioned, which is things like feeling active and feeling alert. And in a paper with Keith Petrie and Jamie Pennebraker and Borgi Sivertson, we looked at this and showed, like we see in all the studies, that positive affect was associated with reduced mortality like we would expect. But when we separate it out and look at the different items within the PANAS, what we see is that this was almost entirely driven by the word active. So feeling active was what was driving this effect. And that if we controlled for active, it essentially wiped out all of the rest of the effects. And this is problematic because we further explored, how tied is this feeling of activity to actual activity. And it really does seem to be very, very strongly associated. In one study that we published in 2020, we showed that people who are in the highest decile of positive affect are 22 times more likely to be everyday exercisers as compared to people who are on the lowest decile. And so these people who are saying that they're active and positive are basically everyday exercisers. And we've since looked at this objectively. I'm going to skip past some slides because I think, am I out of time? Is 12:15 the endpoint? So? Okay, got it.
You're fine. You're fine, Sarah.
Okay. Yeah. Okay, cool. I'm just going to skip past this. So we looked at this with a recent study where we actually measured people's objective activity with actigraphs. We also measured their physical fitness with VO2 max to really look at whether or not the felt activity was tied to true activity. So is it that feeling high in positive affect is associated with, actually being more active and fit and more healthy and potentially more energy efficient, like relevant to Martin's discussion? Or is it that it's really about emotion? And so we did this recently. The study got all sorts of messed up with COVID. So we're just looking at the data now. But what we've shown is that not only do we see the same thing that we saw, where essentially how active you report that you are is essentially, it's a hugely strongly correlated with feeling active. It's an R of 0.5, also related almost as strongly to hours a week of activity. But when we look at objective measures, so are they actually more active? And we look at their actigraphs, we see that absolutely people who are saying that they feel active as measured by the PANAS, that they're taking more steps per day and that they're burning more kilocalories per day. And then even if we look at VO2 max, which is an assessment of it's considered the best indicator of cardiovascular fitness and aerobic endurance, we see the same thing, which is a very strong correlation between VO2 max and feeling, oh, I have a typo in there, but trait levels of feeling active. I'm not sure where that, the ing came from here. So again, we think this is positive affect, but a big chunk of this could just be being physiologically more fit and actually moving around more. Now, I will say there's some good news with the VO2 max, which is that it wasn't correlated with all measures of positive affect. It wasn't correlated with low arousal positive affect, but it definitely is a big part of positive affect, and it explains some of our past studies when we look at that word, I feel active. And so if we look at how much of the item I feel active is explained by non-affect variables, what we're seeing is that age, sex, MHI, VO2 max, we put all these together. I'll get to the MHI in a second. That these are explaining about 38% of variability. And so this isn't really an emotion word. It's really capturing something about our behavior. It's catching something about our objective health. And this matters. So why does this matter so much? It's because if a big part of positive affect, and especially the positive affect that's been predicting health, is because of our health, right? It's positive affect picking up on our fitness, our energy capability, our activity level, then this is just health predicting health. And this is going to have to completely shift how we interpret our past findings, how we interpret our data, and certainly how we conceptualize what we should be intervening on to improve health. And so I think this is just the tip of the iceberg of a really important problem in the field that we need to be thinking about really carefully. Now, given that I followed Martin's talk, I couldn't help but talk about the MHI data that he was discussing and that he's been so gracious to collaborate with my lab on. And I'll mention this in the context of timing and timing mattering. And so, many of us, when we're picking our measures of positive affect, it's very haphazard, right? We're using these big epidemiological data sets. We're kind of putting in whatever we just happen to have access to. It might be how I feel generally. It might be a big broad measure of positive affect. It might be a specific measure of positive affect. It might be how I feel now. It might be how I feel generally. And we're not usually thinking super carefully about what matters. And certainly there's been all sorts of studies showing that even how I feel now can predict longevity. But of course, when we think about what makes the most sense, it's usually going to be a trait measure that predicts our health in the long run because it's how we feel in general. Now, I was obsessed with mitochondria and the possibility that this might be related to positive affect, especially high arousal positive affect, for so long. And I was so excited when I met Martin for the first time at a conference, and I basically stalked him and forced him to collaborate with me because I really wanted to know, is it the case that positive affect was related to mitochondrial function? For example, although I know we're not supposed to say mitochondrial function anymore, but mitochondrial biology. And especially this high arousal feeling, this feeling of activity, this feeling of energy, could it be that when we're saying that, it's just because we're interocepting on our actual energy? And luckily, I don't have to explain how mitochondria work because Martin just did that for half an hour. But when I looked at this, it was awful. I spent so much money, so much time, every blood sample had to be analyzed for like an extra few hours to get this data for like 150 people. And when we looked at this positive affect, unlike Martin's 2018 study, nothing was working. But then when I thought about this sort of nuanced question, and we said, okay, well, maybe it's not trait positive affect, and maybe it's not all of positive affect, because why would calm be related to mitochondria? And we said, okay, well, what if we just look at feeling energetic, which is an item in several positive affect measures? And so if I look at how energized I feel right now, is that associated with my mitochondrial health? And lo and behold, and Martin has not seen this, so this is the first time he's seen this because we're just analyzing this data now. But what we see is that your self-rated energy level, how energetic do I feel right now, is absolutely correlated with how with your MHI index. Which was just, I cried when I found this. It was so exciting. But really interestingly, it's not related to any other positive affect measure. So none at all. So that's really exciting. And it really points again to this need that we need to be thinking about not just the kind of emotion we're measuring, but also the timing. And so if we looked at general energy over a 2-week period, or in general, how your trait energy is, that wasn't related. But when we looked at how you feel right now, that was related to MHI, and that really reflects with the day-by-day analysis that Martin's paper looked at as well. Okay. So I don't have time to get into variability because I think I'm over time now, so I'll skip past that because I'm well over time. But I will say check out Brooke's awesome work on affect variability, which is also showing that it's not just our average affect. So next step, I think what we need to do is admit that with some of these correlations we're finding with exciting new biomarkers, that it may not all be because of emotion, but it may be because of how we're measuring emotion. And that with certain kinds of affect measures, these are likely largely determined, or at least partly determined, by our energy, by our fitness, by our health, and that those are getting picked up by some of the measurement choices we're making. Now, from earlier in my talk, we also need to be thinking about these important moderators like culture. And we just need to be generally thinking just as carefully about our psychological measurements and what questions we're asking, and if we even ask questions versus observe people, as compared to just kind of throwing in our psychological measurements. We need to be thinking as carefully about this as we do our biological assessments. Now, obviously, there's still room for lots of awesome biomarkers that we should be considering. My grad student, Desi Delgadillo, was doing really cool stuff with microbiome, and we obviously heard from Martin and Steve about some exciting other possibilities. But I just want to urge people to not forget about the psych side and to take that as seriously as they're taking their biological measures, because really it does seem to matter a great deal for how we understand these connections. And if we can understand this more deeply, it's going to really help us build better interventions as well. And that's it. Oh, and this is my son, Ian, who woke me up at 4 a.m. and I'm going to blame him for going over in time.
Thank you, Sarah. I'm Wendy Berry Mendes, and I have the honor of talking to Sarah for a few minutes. Also happy to relay any questions in the Q&A. And Sarah, wow, what a terrific talk. And I just want to point out this funny tension when we were designing this day was whether we had you go first or last. And then we started getting all these questions that in many ways sort of predicted your presentation. So I think our conflict or tension about that was right, because you both set the stage, but also sort of helped us wrap up. So thank you for doing that. And I guess I want to start with this arousal piece. I find the data really compelling, and I agree, we often ignore this low arousal positive affect, especially kind of in the US. It's kind of an American thing, high arousal. And so I'm curious, one, you mentioned Jeannie Tsai, sort of ideal affect work, and sort of possibly, the very different desire to sort of achieve more low arousal positive affect in some cultures. And then I'm also reminded of sort of mindfulness-based interventions, which at least to me seem to have a goal of trying to sort of create low arousal positive emotion. So I guess I just wanted to sort of hear you think through maybe the cultural specificity and maybe some of the positive emotion interventions as the goal with low arousal, given your findings.
Yeah, no, it's a really great question. And in the study I only got to briefly talk about, we have a lot of measures of ideal affect, because I think one thing we don't know now is whether or not your ideal matters in terms of your physical health and your physiology, right? So there's one study by Curran and colleagues that used the MIDGA data, which looked at whether or not your ideal affect was associated with, I think it was maybe like heart disease data or something along those lines. And basically showed that negative affect in Asia, where it's not as unidealized, where you don't want to experience it all, it's more accepted, that it's less bad for you. We looked at that at some of the Gallup data, and we didn't see the same effects that you would expect. Basically for us, when we looked at self-reported health, it didn't matter what your country was, it was always associated with benefits. And so, to me, that sort of suggested that regardless of our ideals, there's probably some physiological processes that are making a difference. I mean, when we think about the high versus low arousal, I think a lot of it, what we really need to think about is what we're predicting in the context. One of the confounds obviously we're dealing with is that, it could be that the high arousal stuff is looking like it's good for us, partially because it's representing that we're healthier, in which case, this American desire to feel vigorous and feel excited, like, is this really that good for us? I mean, it's hard to say, because we right now don't have any good interventions to increase high positive affect arousal beyond like sexual manipulations, like showing people sexually arousing things. That's or exercising. That's a good way of increasing high arousal positive affect, which is obviously a huge confound. Right. And so there's been no experimental study where we can look at whether or not, we do this great long-term intervention, make people feel vigor, and see whether or not that improves them. Now, from the opposite perspective with calm, I mean, Jeannie and I talk about this all the time. Like, Americans, we generally don't talk about calm enough. We don't focus on it. It's not our ideal. But obviously, in the case of stress, and especially uncontrollable stress, that's going to be probably what's going to be the most helpful. I have a paper with my former grad student, Amanda Acevedo, where we looked at this in a stress reactivity study and showed that if you have an uncontrollable stressor like pain, and we manipulate your experience of calm versus mid-arousal happy versus high arousal energy, like feeling vigorous, that it was inducing calm that helped in that context. And that makes sense, right? Because you're in this highly arousing, stressful context that you can't do anything about. You're just experiencing pain. Like, adding vigor to that recipe is not going to help you. It's going to make you feel worse, right? Like, I'm full of energy and I can't do anything. But feeling calm is going to help you undo that reactivity. And obviously arousal is a big ingredient in pain. And so it makes a lot of sense that it would work there. And so I think we just need to be better adding to this complexity of nuance we were talking about, in thinking about the context of emotions and when certain kinds are going to be helpful.
Yeah, and that leads exactly to my next question, which is about affect variability. And maybe you just answered this, but sort of rather than always seeking, are people feeling high arousal positive emotion, but that is it context appropriate. Are we feeling calm right before we go to sleep, but vigor right when we wake up? And given that, do you have a sense of how we might best measure that? Say I, have $10 million to do all the best biology. How should I measure emotions in daily life that I'm capturing context appropriate emotions? And is it the roller coaster of emotions that's going to be most predictive?
Can I share my screen again?
Yes. Yeah.
I can show you my one slide on affect variability. Let's see. Are you guys seeing the right thing?
Yes.
Just switch it again. Okay. So if I go back, again, Brooke has been doing this work, not me. So, she can tell you exactly how to measure affect variability. And also my current grad student, Logan Martin, who's doing some great work and has a paper under review on this. Essentially Brooke's been doing this a lot, and so has Anthony Ong. He's got some great work on variability. And what most of these papers seem to be showing is that, over and above your average, variability adds something. So that's good for us to know. In this case, variability was bad. So positive affect variability, if you're high in positive affect on average, but then you add that variability piece where what you're saying is you're like, we're going up and down, that was associated with worse immune function. In this case, it was antibody response at 1 month and 4 months after vaccination. And so I don't think that roller coaster is good, at least for positive affect. For negative affect, you could imagine that it could give you a break. And so if you're high in dispositional negative affect, but you're going up and down, that could be better. But for positive, I mean, as much as we talk about heart rate variability and how variability is good for us, for positive affect, if you really are on a roller coaster, that may not be buying you much. So was your question like, how do we measure?
Yeah, so if that's the goal, is to capture that variability, if it's context specific, how best to approach that?
It's a really good question. I mean, because I think you need I think all of the technology that's being developed now to do real world monitoring, and we know where you are physically, we know the noise around you, like combining some of those tools with biosensors so that we could kind of hear where you are and see what you're doing will get us to better understand this. In terms of how to best capture the variability piece, I don't know, because I'm not even sure. I don't think we really know yet the physiological mechanisms underlying what variability is doing, like why variability is adding. Like, is it this lack of restoration? Is it giving us restoration? Is it, certain kinds of autonomic function? So I think it's just going to be, using a lot of big data to really collect as much as possible so that we understand what kind of emotion helps us when. One thing we're looking at is in my lab with this recent study is different kinds of stressors, because oftentimes we're assuming all stressors are kind of equal, and obviously they're not. And so I think it's just going to be collecting as much information and really looking for sort of the matching, like what is the ideal match? Is it good to be variability? Is it good to be high in variability if you're in this chronically stressful context? Because maybe that gives you a break. If you're in this great stable environment, maybe variability is not good for you, but we just need more data. So I think just collecting a lot of data, just as, thinking about all of these variables of, kind of stressor, kind of emotion, what kind of arousal, what kind of biomarkers are changing, so that we can put all that together and try to understand it. But yeah, it's really hard.
Great. Well, thank you so much, Sarah. There are a few questions for you in the Q&A that we hope you can respond to. And with that, thank you so much, Sarah.